This is the last part for chapter 27. We're going to look at recombinant DNA. So you need to understand that this means that the DNA that contains two or more DNA segments not found together in nature. So that means we're going to combine the DNA of two organisms that will not be able to reproduce if they were next to each other. One of really the key points for DNA recombinants is the use of plasmid. Plasmid are extremely easy to isolate. They exist in those bacteria, and they are just a circle of DNA that exists, and they can replicate through the normal base pairing pathway that we know because the genetic code is universal. How do we do this? Well, we use restriction enzyme. Those are the scissors that we use for recombination of DNA. Now, what they can do, they are just going to act in the sequence of both strands of the double-stranded DNA and still able to read in the same 5' to 3' direction. So what's going to happen is that when you use those restriction enzyme, we call those like this, they will just cut the DNA and leave some parts that we call the sticky ends, because those are going to be able to match up with the bases that are complementary sequences. So how do we do recombinant DNA? Well, you cut the two DNA segments to be combined with that same restriction endonuclease, and you get here DNA fragments that are complementary each other with those sticky ends. Then you mix both of them in addition of a DNA ligase enzyme that will help to join them together. Remember, the DNA ligase is the one that is going to link together the Okazaki segment, and we talk about this when we talk about the DNA replication, the one on the lagging strands. And well, this is going to be really interesting to see the end product. So here below you have a diagram showing you a chromosome from the organism of interest, and then you see the plasmid of a bacterium, bacterial plasmid on the left. You are going to use the restriction enzyme to just cut both the plasmid in the bacteria and the gene of interest that you have on that chromosome. Then you use DNA ligase and you can have what we call a recombinant plasmid. So that altered plasmid is back inserted into the bacterium cell, and you just keep and let it go through transcription, translation, and synthesis of the protein that was coded by the section of the gene that was inserted. The advantage of bacteria is they multiply very, very quickly, and then you get a very large number of them. All of them contain the recombinant DNA, and they all make that protein that was encoded by that portion of DNA, the gene that was in that other DNA. So genomics is the study of whole sets of genes and their function. Those are here some examples, what we know, where we use them. Those are fields of study such as biotechnology, bioinformatics, functional genomics, comparative genomics, proteomics, pharmacogenomics, and pharmacogenetics, as well as toxicogenomics, genetic engineering, gene therapy, and that leads to ethical implication of how knowledge of the human genome is used, and that's what we call bioethics. So that leads us to the GMO, genetically modified organism that can be both plants and animals. Well, those plants or animals have been processed for centuries, and that's what we use to have natural selection, then artificial selection, and we were able to hybridize few known varieties. But then things change when we can actually include a gene of a very different organism and help us generate crop plants from animal which are going to be able to display desirable characteristic and also removing the genes we don't want to have. So going back to GMOs, well, we use them, and actually more than 80% of everything we have here in the United States is going to be genetically modified organisms, mostly for plants, corn especially. And then if you look at this, well, tons of corns were destroyed by the European corn borer. And they found a way to solve that problem, and finding here a gene in a bacterium, we call it BT, Bacillus thuringiensis, and it was able to be transplanted into the corn, and it will make the corn to be able to produce a toxin that will kill the caterpillars that will just normally invade them. Now, there's a lot of tests that are underway with GMOs, talking about coffee beans, they are caffeine-free, potatoes that can absorb less fat when they are fried, and the famous golden rice, which is a rice that provides vitamin A, which is needed especially in poor population where there's a lot of death, blindness that are linked with insufficient vitamin A. So those are positive aspects of those. But no, we have a problem right here. Well, will those genetically modified organisms intermingle what natural variety normally takes place and cause harm to them? No, we also have this question about should we have labels on the food that tell you if those are genetically modified? And this is like another area, and you have to think about it. We also don't really know if those harmful substances could enter the food supply, and there's a lot of debate that exists. And we have here one example, the non-GMO project, which is to offer to consumers non-GMO choice for organic and natural products. No, there was really a lot of debate about labeling those genetically modified organisms. All I can say is that myself, if I go to a store, we don't have necessarily labeled GMOs, although things have changed. But the way they changed this is that unless you have a way to read the code bar on a product that will tell you if it does contain GMO or not, my approach is a little bit different. Everything that has a label that says non-GMO, that's why I buy it. Okay, talking about gene therapy. Well, this is very important because we are looking at here a disease-causing gene that can be corrected or even replaced by being able to insert a functional, healthy gene. This is here some of the expectation is still kind of in trials, and we're looking at here treating monogenic diseases, and those are just the result of a defect that exists of one single gene. That leads to gene therapy, where we're trying to find non-pathogenic viruses. Those viruses will act as vectors, and they will be able to replace the way viruses work, therapeutic quantities of a DNA directly into the nucleus of one cell. Well, we hope this method could help in helping to eliminate lifelong and inherited diseases, and there's a lot of studies that are ongoing. Expectations remain greater than achievements so far, and the FDA, the Food and Drug Administration, that was in 2014, has not approved any human gene therapy product for sale. You also need to think about what we can actually do with personal genomic survey. So if you have a patient that lacks an enzyme that is for any kind of drug's metabolism or has a monogenic defect, you could actually tailor individual therapy. Cancer therapy, for example, understanding the genetic difference between normal cells and tumor cells could actually help with the chemotherapy. But at the same time, well, if you look at this, genetic screening of infants will help people to use gene therapy to eliminate threat of monogenically diseased or even some lifestyle adjustment for an individual who has SNPs that can actually let us know he has a risk to have a disease or may be susceptible, and that will help for combination of genetic and environmental influences. But at the same time, that raises an issue with what we call bioethics. Well, if we have this genomic revolution, this is good to help patients, but at the same time, well, we may have social implication. Now think about it. Well, what about if you're the person who hires you, if your employer finds out that you have a genetic disease that will make you not able to work within 20 years? Would they have access to this? So to help kind of find solution about that bioethics, there is a ELSI program. The National Human Genome Research Institute deals with ethical, legal, social implication, so ELSI, of human genetic research. And the scope of ELSI is actually very broad and make people think about it. Who should have access to personal genetic information, and how will it be used? Well, what about if your healthcare provider has access to your genome and finds out that within 30 years you most likely will have a cancer? Will they have a chance to deny you for any benefits? So this is really bringing some ethical issues. And who should own and control that genetic information? So also, other questions, should genetic testing be performed when we don't have any treatment available? Well, can we also rename disabilities as diseases? Can we cure them? Can we prevent them? So there's a lot of things that are going on here in the bioethics. And then preliminary attempts at gene therapy are very expensive. Now, this is bringing another question. Who will have access to those therapy? And then who will pay for those? So we also need to think about, can we re-engineer the genes of parents who are defectuous so we pass on to our children only the good genes? So it really reveals, it's kind of, everything we have in science, there's two sides. One of them is really good. We can help patients. We can help people maybe take steps so they don't have to worry too much about potential disease if they take any steps. Avoid suntan if there is cancer, skin cancer in their family, and we find out that they are really more prone to have the skin cancer. Now, what about if there is no therapy? What about who is going to use that information? Can it be just the person who gets information about his genome, or is it going to be passed on to healthcare providers or your employer? So that's kind of one of the things I would like you to read about that chapter and make you think about the good thing and the less good thing about the development of that gene therapy and the technology that we have.